Bismillahirrahmanirrahim. Today we will discuss about the mitochondrial structure and function. So we say in eukaryotic cell, especially we are discussing mitochondria in eukaryotic cell because in prokaryotic cell there is no mitochondria. So in eukaryotic cell, mitochondria is a site for link reaction, T.CA cycle, and oxidative phosphorylation. So as a result of glycolysis, pyruvate was produced, pyruvate was converted to acetyl-CoA. So this acetyl-CoA was formed in the link reaction, then this acetyl-CoA which was formed in the link reaction sent to the T.CA cycle. So right over here, this acetyl-CoA again decarboxylated through the process of T.CA cycles to give us NADH and FADH. So NADH and FADH were produced by the process of glycolysis, by T.CA cycle and link reactions. So now they are used up in oxidative phosphorylation to give us ATP. So we say mitochondria is a site for link reaction, T.CA cycle and oxidative phosphorylation. Now we say in each prokaryotic cell they contain a mitochondria. So number of mitochondria depends upon the activity of the cell. So different cells of the tissues, so they have the different number of they have different number of mitochondria depending upon their activity. In case of hepatic cell, like liver cell, contain 1,000 to 2,000 mitochondria in each cell. So we say mitochondria occupy 25% of the volume of the cell. Now we discuss the structure of the mitochondria. If you say function of mitochondria, including link reaction take place inside the matrix of the mitochondria, T.CA cycle take place in the matrix of mitochondria, oxidative phosphorylation take place in the inner membrane of the mitochondria. Now we discuss the structure of the mitochondria. So mitochondria is a double membrane bound structure. So in valve of the mitochondria is double membrane. So one of them is known as outer membrane and other one is known as inner membrane. So the composition and function of outer and inner membrane, they are absolutely different. Outer membrane is smooth and more permeable for the small molecule. Inner membrane folds to form cristae. So these cristae increase the surface area of the mitochondria. So the more multiple enzymatic reaction take place at the cristae, so increases the surface area of the cristae. We say the double membrane, inner membrane, and like say outer membrane and inner membrane form cristae that increases the surface area of the surface area for multiple reactions. So at the inner membrane, electron transport chain or oxidative phosphorylation would take place as well. So synthesis of ATP also take place by the ATP synthesis enzyme which is present in the inner membrane. Now we say is the outer membrane of the mitochondria, then inner membrane. The central area of the mitochondria is known as matrix. So matrix is the region where link reaction, T.CA cycle, and oxidative phosphorylation would take place. And inner membrane, right over here, the inner membrane, proteins are embedded. They form the complexes of oxidative phosphorylation where electron transport chain is situated. So it would give us hydrogen ion gradients. So there is a hydrogen ion gradients between between the matrix and intermembranous space. So intermembranous space is the area between the inner membrane and outer membrane. So because of this gradient energy is produced. So that energy is used up for the formation of ATP at the matrix by using ATP synthetase enzyme, by utilizing ATP synthetase enzyme. Now we say double membrane structure of mitochondria, outer and inner. So they have the different composition and function. So outer membrane is more smooth, inner membrane folds to form cristae. So this inner membrane folds to form cristae, and the number of the cristae depend upon the activity of the mitochondria. So these cristae are densely packed and they are large in number in more active mitochondria as compared to inactive mitochondria. We say active mitochondria would have more complexes, more cristae, which are densely packed. They are very close to each other. Now we say ATPs are formed in the matrix. So ATP synthetase are used for the formation of ATP. So hydrogen ion gradient between the inter and inner membranous outer and hydrogen gradient is formed between the matrix and intermembranous space. So hydrogen ions move inside through the ATP synthetase to give us ATP. So ATPs are formed at the matrix. Now we say we have the different multiple structure we can see inside the Inside the matrix, so one of the structure is known as ribosomes. So it also contains ribosomes. So these are 70s ribosomes which are present. These 70s ribosomes are present in the matrix of mitochondria as well. So mitochondria distinguish another structure as well. So it has its own DNA as well. It has its own circular DNA and its own mitochondria, like say its own ribosome. So we say mitochondria is known as semi-autonomous body. So we use the word semi-autonomous body because it can carry out certain of, like say it can fulfill partially its need as well. So we use the word semi-autonomous body for the mitochondria. Mitochondria has its own ribosome, which is 70th ribosome. So 70th ribosome makes it resemblance with prokaryotic cell. It has its own DNA. So this DNA is also similar to the DNA of prokaryotic cell. So there is an association between the mitochondria and prokaryotic cells. So that association was explained by endosymbiotic theory. Right? So endosymbiotic theory explains the origin of mitochondria, or we can say the evolution of mitochondria from the prokaryotic cell, right? So here mitochondria has its own 70s ribosome in the matrix. It has its own DNA. So these two structures, mitochondrial outer membrane, also the porine. So these structures, like say, give us the clue that it was evolved from the prokaryotic cell. So in the beginning of the life, we say 3.8 million years ago. So there was only prokaryotic cell on the earth. So this prokaryotic cell developed to form a initial eukaryotic cell, which is known as primitive eukaryotic cell. So prokaryotic cell was evolved to form primitive eukaryotic cell. So when the size of this eukaryotic cell would, like say, keep on increasing, the size of the eukaryotic cell keep on increasing. So as a result of this, there the surface area to volume ratio of eukaryotic cell would let's say word decrease. To overcome this issue, because by increasing the size of the cell, the surface area and volume, surface area to volume ratio, which is concerned with the absorption and excretion of the cell. So surface area to volume ratio was reduce. To overcome this issue of surface area to volume ratio, because it was going to be reduced by increasing the size, this cell membrane folds to form small folds. These form small folds. So later on, these folds were pitched off from the cell membrane in containing the central DNA only. Containing the central DNA. So now this central DNA was surrounded by the folds, obviously surrounded by the small vesicles which were pitched off from the cell membrane. So right over here, this primitive eukaryotic cell, it engulfed this primitive eukaryotic cell was a bit evolved or developed. This cell would engulf an aerobic bacteria. So as an engulfing of aerobic bacteria, so it, like say it start utilizing this aerobic bacteria for the formation of energy. Right? So when this energy was being formed with aerobic bacteria, so on the other end, this cell also engulfed in cyanobacteria. So this cyanobacteria was providing the food for this bacteria, for this eukaryotic cell. So this eukaryotic cell, primitive eukaryotic cell, engulfed an aerobic bacteria. This aerobic bacteria modified to form is further, like say, modified to form mitochondria. So we say this mitochondria is evolved from the prokaryotic cell. So there are the certain points which, like say, which follow this theory, or which support this theory. We can say this DNA of the mitochondria is similar to DNA of prokaryotic cell. It has a 70th ribosome, and this 70th ribosome is also present in the prokaryotic cell. And also, there's a very important thing: this, like say, mitochondria would multiply by the binary fission. So binary fission is also exhibited by prokaryotic cell. Another thing is that this membrane of the mitochondria, outer membrane, contain porins. So these porins are also present in the prokaryotic cell, in the membrane of prokaryotic cell.